Okay, a couple of things that I just wanted to alert you to. That I posted. I actually posted let me go back here. I posted the study guide on a module on Canvas that is called lecture study guides. So have you been able to see it? I mean, I just posted it moments ago, so. But the study guide for exam one. And the module's name is Lecture Exam Study Guides. Actually, it may be okay for us to go through that quickly. Let me just show it to you. I also wanted to record, so that's one of the things I'm doing up here. So let's see if it actually works. So this is should be very familiar to you by now. And of course, this is our platform for the exam as well, the PowerPoints, the lectures, etc. So, the lecture recordings are here. And let's see where our lecture exam study guides are here at the bottom. Okay. So far there's only one. If we click on that, it should open. So let's just go through a little bit about some strategies for the test. The test will be here a week from today. And what you need is a pencil. So that's the basic equipment that you need to bring. So the exam is mostly multiple choice. So I will provide the bubble in sheets. Okay, so what you need is a pencil. Optional is a piece of 11 by 8, a regular sheet of paper, 8.5 by 11 with notes on one side. So you're free to provide yourself with a little cheat sheet or whatever you want to call it, exam support. So that piece of paper can be handwritten, it can be typed, you can draw on it, you can cut and paste, you can do anything you wish. You will hand it in, so you probably want to be somewhat professional in your presentation. But I mean, what I mean by that is don't put any graphic swear words or anything. I guess even that's fine, I guess, whatever you need. But I will be looking at it, is what I want you to know. So you'll hand it in with the exam, and what I require is that your name be on it. So put your first and last name on the whatever you bring with you. Are you clear on that? Okay, good. I suggest you get here early, like a few minutes early if you can. It just saves yourself stress. We will start on time. I'll be handing out the exams at 1 p.m. And so if you're already in your seat and ready to go, that just helps. I want you to have the entire time. Okay? So, so no phones, no shenanigans. Okay. In technical terms. Okay. So obviously you will keep your eyes on your own paper. I'll be here, so I know I can see your eyes. So just don't. And then we don't have to deal with it anyway. You are training to be ethical professionals. So you're not going to cheat. Um, so this is the study guide. I gave you some suggestions. You can read these yourselves. This is what I would do to study, but I'm not you. So you need to do your own. To get things in your brain. Uh, Certainly, I guess you could use AI to create a study guide for yourself, but I mean, you need to engage with the material to learn it. So however you do that, I think drawings are really helpful. I like colors, I like stickers. So, I would have some kind of notebook or pieces of paper. In fact, what I recommend is that you take a piece of paper that is blank for each topic, and I would write at the top, plasma membrane. And then really it helps if you write even before you start to study. What do you remember? What do you remember? What is the basic structure of the plasma membrane? How does that relate to its function? Those are questions that we're going to ask for everything we do, right? What is the structure? What is the function? Can you draw it? Do you remember the details? It's what you don't remember that you want to study, right? So then you can fill in the paper with the details from the PowerPoints, from the modules, and from the book. Okay, so the book has sections on each of these topics that we're covering, and that I would use all of those resources and organize them. So I just provided some ideas for you to think about. Every topic that we talk about, practically these three big ideas come back, right? Biological levels, structure relates to function, human bodies are the product of evolution. These are some of the things that we talked about in the initial parts of the class, an introduction to anatomy. What is anatomy? Could you define it? What are the goals of studying anatomy? What is comparative anatomy? What are some important, historical things that happened that help us to understand anatomy? And one that I highlighted was the discovery of the structure of DNA. And think to yourself, why would I choose to take time, our valuable time, to talk about that? So why is that important to our understanding of our own bodies? That's a good question to always ask. Why is this important for me to understand how my body works or how I'm structured? The one thing that we did specifically was I had you do that little exercise. I don't even know if you remember it, but when we were here together, you took pieces of paper and you chose a body system and tried to apply the biological levels to that. So what I would expect you to know, and we didn't go over it in huge detail, but I think you should, is I would want you to be able to list the body systems. There are 11, so make a list of 11 body systems. What is the main function of each of those systems? And what are 2 or 3 primary organs? Okay. You could even make a little table for yourself. Body system, digestive system. What is the main function or the primary functions of the digestive system? What are 2 or 3 organs in the digestive system? I think you could do this from memory right now, probably. But to review, I suggest that you go to 2 pages in the textbook, 8 and 9. And it's that diagram, that same diagram that I believe I put in our PowerPoint. So it was a picture of the 11 body systems, and it's just a really quick and dirty review. But that provides a foundation for our discussions as we go forward. So I think that's important for you to know. The evolutionary context for anatomy, this is just a review of the lectures that we talked about. What are some of the events? What were the things that happened? Why is this important to us? Because we're founded on cells, for example. So that really dates back to the earliest life on Earth, our ultimate early ancestors who gave us those fundamental features of ourselves. So I gave you some specific questions to give you a framework for putting some of those details into some kind of organized fashion. And then cells are what we're talking about now. And then the next topic is tissues. We're going to get to tissues by Tuesday. So the material on Tuesday will be on the test. So you may want to start reviewing that module this weekend. Hopefully we'll get to tissues today. So that's kind of the plan. There will be one short answer. You know, you've been doing multiple choice examples because you've been doing the Connect quizzes, correct? So it shouldn't be a mystery to you what kinds of tests or what kinds of questions are going to be on the test. I really have no intention to be tricky. I very much intend to be challenging. So I don't want to trick you. I want to know that you have engaged with the material thoughtfully. I want to know that you have learned and understood something, and I want you to feel proud. When you get those questions right, just think of how much you have already learned. So, and it's not trivial. You know, I try not to waste your time. This is material that I think will stand you in good stead as you go forward. So the multiple choice questions are what they are. And then you will have an opportunity to do one short answer question. Typically what I do is I give you choices. So there might be 3 different questions and you could choose the one which you felt you can answer best. Okay. These are examples. So if you answer all of these, you should have a good handle on what would work for you in the test. Okay. So these are worth 10 points. It's the difference between one grade and the next because the whole exam is worth about 100 points. In fact, the exam is worth 100 points. So, so this just gives you an opportunity to practice and be a little creative about what you've learned. So to speak, sorry, I should never use that word. But are you understanding, clear, comfortable? Even I am not old enough to use the word groovy, so I just want you to know that. Okay. How are you doing? I just realized I had a lot of coffee this morning. So maybe I pounced a little bit too strongly there, but you're okay? Yes.
I was noticing some, I guess, tech discrepancies on Canva. Because it's on the APR set on that Connect portal.
Yes.
It's not reflecting as submitted as Canva, it's affecting my grade.
You know, I don't know what the problem is. And so what I would say to you is that it's not affecting your grade. It is affecting the scores that are on Canvas. So as long as the score is in the Canvas column, I will figure that out. Yeah, you're not the only one with that problem, and it's something to do with Canvas this semester that I just haven't worked out yet. I never have had that happen before, but I haven't had a chance to meet with the people who can figure that out. So let me assure you again, I think I know what you're talking about. and you're not the only one. But on Canvas, is your Connect score in the grade book?
It's in the grade book, but.
That's what's important. That's what's important, because that's a matter of me finding out what key to press or what button to press in Canvas. Okay, so from my point of view, as long as you have done the assignments, we'll figure out the grade. You can add up the points yourself. So the possible points are whatever, and you can figure out your percentage. Your grade is not affected. I do want you to know that and to be assured about that. As long as it's in Canvas, we'll figure it out. Okay. So I know it's a little frustrating, but it's a minor glitch. And it's not your fault. And I don't think it's my fault. So, but we will figure it out. Do you know what she's talking about? Maybe it is just a few people, but you are not the only one. So I hope the rest of you are okay. But I will figure it out. So not to worry as long as you've done the work. Okay, let's move on to our discussion. Sure. Okay, good. All right, I wanted to make sure it was right for the recording. Okay, so the topic that we had stopped with last time was the active transport. If I remember, I mean, we were talking about transport across the membrane. And we were trying to figure out why this might be important. So far we had talked about diffusion of molecules and ions. Ions are electrically charged particles, like sodium and potassium are electrically charged particles. They are called ions. But the diffusion of particles, whether they're molecules or ions, is important biologically. An example that I would use, I think that I use for diffusion is the movement of oxygen and carbon dioxide across the membrane of the teeny little cells. This is representing an air sac in the lungs. When you breathe in, you breathe the air from the environment in through the tubes that lead to the lungs. And at the microscopic level, there are millions of teeny little grape-like sacs. And those sacs are lined with very thin cells. The black dot represents a nucleus. Each cell is shaped like that. And what happens is as you breathe in the oxygen, it flows into those air sacs. It is a very tiny little molecule, and it can diffuse through the phospholipid bilayer. And ultimately, it will go into the blood vessels, the little teeny blood vessels that surround the air sac. So it diffuses from areas of high concentration to areas of low concentration. In contrast, when you have accumulated carbon dioxide in the blood, the CO2 will go the opposite direction from the blood into the air sac. And the way that you get rid of it is that you exhale. The CO2 diffuses across the plasma membranes into that little air sac and then out. And that is an example of how important diffusion is. Osmosis was another type of passive transport that we talked about. Do you remember one reason why osmosis is important in our body? We talked about a couple, but do you remember something about why osmosis might be important? Yeah. Absolutely. It is a balance of water, right? Between like the inside and outside of cells. Absolutely. And so if you do not have a balance of water molecules on the outside and the inside of the cell can change shape, and that can lead to big problems, right? Including even the bursting of the cell, which means that the cell dies. So osmosis helps you to maintain fluid balance. It helps you to maintain the balance of water on different sides of a compartment of an area, including the cell. So if you don't, you have problems. So good, that was a good summary, thank you. So we had begun to talk about active transport processes, and active transport requires ATP. That's why it's called active. So ATP is the energy that cells produce in the mitochondria. An example of active transport that we talked about very briefly was the sodium-potassium pump. So let's take a look at this just in a little more detail. So what are you looking at here? What are the golden sort of structures right here? Phospholipids, okay? So this is a very small part of the plasma membrane of one single cell. So, so if this is the cell, this is the plasma membrane. So in terms of scale, that picture is representing a very small piece. And then what it's showing is the protein channel that extends through the membrane. And that protein is where ions, electrically charged particles, move back and forth. Two that you should know, that you should be able to recognize, are sodium and potassium. So those are notations that we use all the time in both anatomy and physiology. So what this is showing is that this particular protein in the membrane can power the movement of sodium against its concentration gradient. That's why it requires energy. So even though there's already more sodium out here, you're going to push even more out. And even though there's more potassium in here, you're going to push even more in. And that requires energy because it's not going in the natural flow of things in the way that they want to flow. Thank you for that. So, yay. Oh, good. Perfect. Yeah, that's great. I really appreciate that. So this will be fun. So we're gonna do a little activity that Chelsea is looking at in a while. So don't get too excited yet. So this is the sodium potassium pump. We said, why is it important? because it is necessary for nerve function, for skeletal muscle function and movement, and for cardiac function. Okay, so heart function. So if the balance of sodium and potassium across the membrane gets messed up, that is called an electrolyte imbalance. And that can lead to all kinds of things, including heart arrhythmias, a funky heart rhythm. Okay, so our sodium potassium pump is very important to us. And I think one little fun fact that I mentioned to you the other day, do you remember what I said about it? That I think is really startling, but you may not remember or know or be able to read my mind. It requires about half of your calories every day to provide the energy required to move sodium and potassium back and forth in the cells of your body. So, so that is an example of active transport. It requires energy. Another example of active transport that we briefly mentioned the other day is phagocytosis. This is a form of endocytosis, which means bringing things into the cell. But specifically phago, that root word means eating. So this is literally the cell engulfing things and pulling it into itself. And the major example is white blood cells that are able to change their shape and encircle bacteria and bring them into themselves, and then they digest the bacteria and get rid of them. So this is a major way that your body rids you or protects you from infectious disease. Question? Okay, so this is a picture of a white blood cell that is engulfing bacteria, bringing it into itself, and then it will digest it and break it apart. That's pretty cool. So this is the opposite. This is called exocytosis, and this is where you get rid of something from the cell, or you export something from the cell. So phagocytosis is bringing it into, exocytosis is getting it out of the cell. So often exocytosis involves the production inside the cell of little bubbles, which act like packages. I mean, essentially it's a package that the cell produces, which is called a vesicle. So these vesicles themselves are phospholipid little bubbles that are produced by an organelle inside the cell, which is called the Golgi apparatus. Let me see if I have a picture for you. Yes. So this is looking at, let's again orient ourselves here. So here is the plasma membrane. So this is only one portion of the cell again. So you're looking at a little peek of the nucleus here in the middle. You're looking at an organelle that's called the endoplasmic reticulum here. And then you're looking at this stack of pita bread type situation, which is in green. And that is the organelle that is called the Golgi complex or Golgi apparatus. The Golgi complex produces bubbles, membrane enclosed bubbles that are called vesicles. They're called secretory vesicles, sometimes even more specifically because they secrete substances or export it. So the secretory vesicles or vesicles are little packets that move toward the membrane. Because they are composed of phospholipids, they can fuse with the plasma membrane just like two soap bubbles can fuse together. And then when they do, they can release whatever it is they have inside them to the outside of the cell. So this is a way that a cell can produce something that you might need in other parts of the body. it packages it and releases it. Produces, packages, releases, okay, through the process of exocytosis. You with me? Okay. An example, let's do a specific example. You have an organ which is called the pancreas. You know the pancreas? So the pancreas is in the abdomen. You'll see it in the cadaver. It's a cruddy little organ. It's not my favorite, but it is. Ugh, it's underwhelming. But it's very important functionally. So the pancreas has cells that produce a protein, which is something that you may be familiar with. That protein is called insulin. Okay. Insulin is a hormone, and it is also a protein. So structurally, it is a protein, and it acts as a chemical messenger. So, and specifically, it is released into the blood, and so it is called a hormone. So if this were a pancreatic cell, what you would see is that there are, this central circle is the nucleus. Inside the nucleus, you find DNA. Would you agree? Inside the pancreatic cells, there's a part of the DNA that would be activated that codes for or gives a message for building insulin. What would you call this portion of DNA that gives a message for one protein or part of a protein? Do you know? A portion of DNA that codes for a protein is called a gene. So that's a section of DNA that gives a message to build a protein. So this is a whole process, right, to get stuff done in the cell. So the gene would be activated. Here you have a structure which is called the endoplasmic reticulum, which we'll talk about. But it has the machinery to put together the protein. And so we're gonna make this a red protein, I guess. So you're gonna make a little chain of protein that is insulin. That is sent to the Golgi apparatus, which is the pita bread type thing. And it produces the bubble, the vesicle that can package the protein. And then the bubble floats to the plasma membrane and allows that insulin to go out of the cell into blood vessels, which then carry the insulin to every cell in the body. When the insulin reaches those cells, they respond to it. Do you remember the functions of proteins in the membrane? Some of them act as receptors. So there are receptors on cells for insulin, which is a cell signal. And then the cells in response allow glucose to enter from the blood. Yeah. What if you're insulin resistant, it means that the cells will not respond to the message. So this is such an excellent question. So insulin is a hormone that regulates blood sugar, right? How does it do that? It opens the door in cells throughout the body to take in glucose from the blood. So you decrease blood sugar levels by importing it into the cells of the body. And then they can use it to produce energy, right? To produce ATP. Okay, so if you're insulin resistant, your pancreatic cells may still be producing insulin. The insulin is traveling in the blood, but when it reaches the cells, for some reason, they are not responding to it.
human growth hormone. Does human growth hormone help with its resistance?
Yes, it's a complicated story, but yes, totally. Okay. So, so exocytosis, that was a long involved example. But exocytosis is the ability of cells to package material and export it for use elsewhere. Insulin is the example that we use to illustrate exocytosis. All right, so let's look at some of the other parts of the cell. So the nucleus is sometimes called the control center of the cell. You probably have heard that somewhere in your past. But why is it called the control center? Well, it contains what? DNA. Okay. So DNA is the control of the cell because it tells the cell when to build proteins and what proteins to build. So basically it controls a process which is called protein synthesis. Protein synthesis is the ability to build proteins. And proteins do everything in the cell. They provide structure for the cell and they provide the platform for cellular processes. They're necessary for all life processes. So the structure of the nucleus is that it has a nuclear envelope, which is also composed of phospholipid bilayer. But the nucleus is a little bit weird because it has a double phospholipid bilayer. So it's like a club sandwich. It has two layers of bread. Yeah, there you go. And then you'll notice that there are these little red dots in this picture. The red dots are nuclear pores, they're called. So they're little holes punched in the nuclear envelope. And the nuclear pores allow for the movement of very specific substances into and out of the nucleus. So that's important. Its structure is important because what it enables to flow in and out is it does not enable the DNA to leave. So it prevents the DNA from moving out of the nucleus. The DNA never leaves the nucleus. It is locked in. But there are partner molecules that can leave, and the partner molecules are RNA. So what can flow in and out of the nuclear envelope is RNA. And RNA is a partner that is structurally similar to DNA, but it does all the work. So it carries out the instructions of DNA. Similar to the cytoplasm, the nucleus is full of a gel-like liquid, which is called nucleoplasm. And then inside, there is an even darker area under the microscope that is called the nucleolus. And the nucleolus manufactures RNA and ribosomes, which are another organelle that is involved in protein synthesis. So if you look at the nucleus with an electron microscope, you can see the nucleolus, the plasma membrane. This is the surface of the nucleus. Looks like kind of a moonscape that shows the little nuclear pores. So the mitochondria are another organelle. They are sometimes called the powerhouses of the cell. You heard that? So the reason is that they produce ATP. ATP is a molecule that carries energy that the cell can use for work. So you transfer the energy from the food that you eat to ATP. Interestingly, I keep talking about the DNA as being in the nucleus. And the vast majority of our DNA is in the nucleus, but the mitochondria is the only other organelle that has pieces of DNA. So the DNA that is found in the mitochondria is called mitochondrial DNA, or mtDNA for short. Why would these little weird mitochondria have their own DNA? It's a really interesting question. And the reason is that billions of years ago, mitochondria were once a separate organism. So they were once a bacteria that lived on their own. So they had DNA just like all bacteria do. But sometime over 1 billion years ago, they were incorporated into other cells. They were like hijacked by other cells because of their ability to use oxygen to produce ATP. And so the larger cells that engulfed them became eukaryotic cells. So eukaryotic cells have mitochondria and a nucleus. So the mitochondria being incorporated into these cells was a huge, 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 huge evolutionary innovation for us. We are eukaryotic organisms. We are built out of cells that have mitochondria. And that's important because it gives us the ability to use oxygen to produce ATP in the process of aerobic respiration. So aerobic respiration is the process of using oxygen to produce ATP. Why do you think that's important to us? Well, how long can you live without breathing? How long can a person live without breathing? Does anybody know? What's your guess? Let's take bits. Yeah. What'd you say? Okay, 8 minutes, about 8 minutes. Anybody else? There's no other bids besides 8 minutes. 5 minutes. 3, 5. How many? 4. I'd say you're all correct. Okay, it really depends on the person. I'd say most of us, the edge would be 8 minutes. But do you know that there are people that I think that the world record was just broken last year? And there was a person who held their breath for 17 minutes. And have you heard of free diving? So they're crazy, right? But it's easier in cold water. It's easier in cold water because all of us have this weird mammalian reflex. When we go under the water, we automatically slow our heart rate, which increases your ability to sustain your orbits for longer without taking as much energy.
Which cases worth like an hour and a half of ice.
Exactly, the cold water helps to preserve you, so there are those really weird places where people are falling into like an icy, through the ice in a lake. And you think, no chance that they would survive, they pull them out and they have like gone to torture or something. Free divers can, again, I think it really demonstrates the ability of our minds. If you practice enough, you can, and I don't recommend that you try this, but these people are like world-class athletes, so I think the record now is like going down free diving without any scuba gear or anything like. Well, people now go over 300 feet deep in competitions. Sometimes their eyeballs pop out when they come on, but they do it. I mean, because the pressure is so great. But you can train yourself. But just think of the mental effort it would take to overcome the fear. I mean, so they actually sometimes they go down on weighted sleds so that they can get down more quickly. And then they have to come up on their own power. It's dark down there. So they really have to, I think you have to force yourself not to panic. And that would make me panic, right? So, but anyway, it's incredible. Well, but for most of us, I think 5 minutes would be asking for a lot without oxygen. And I went down that rabbit hole. But the point is, don't we need to breathe? Yeah. Why do we need to breathe? Because of mitochondria. So the fact that eukaryotic cells hijack those little guys means that we have a respiratory system. So much of our anatomy is based on the ability to use oxygen to produce ATP. Our respiratory system is designed to take in oxygen, right? And to exhale a byproduct of aerobic respiration, which is carbon dioxide. We have to get rid of the carbon dioxide. Our circulatory system transports the oxygen to every cell in the body so that those cells can efficiently produce ATP. Right? Our digestive system is designed to allow us to break down nutrients into, for example, glucose. You eat cereal in the morning, you're going to break that down into molecules of glucose that can be transported in the blood. Under the influence of insulin, those will be transported into cells. The cells will use the glucose in the presence of oxygen to produce a lot of ATP. A lot of your body is based on the fact that over 1 billion years ago, some big greedy cell hijacked these little critters. I know, it's so incredible. And the other thing that makes this so interesting is that your cells have mitochondria in it, right? A muscle cell, a skeletal muscle cell, for example, has a lot of mitochondria compared to other cells, right? Because we need the ATP to fuel our movement when you run or bicycle or whatever it is you do. How do you get more fit? Aerobically. How do you get more fit? You exercise more, right? Are you going to develop more muscle cells when you exercise more? Muscle cells don't reproduce once you're an adult. They very rarely can you make more new muscle cells. But you can increase the number of mitochondria because they have the ability to divide on their own. So when you stress yourself aerobically, when you start a fitness program and you think I'm going to go out and I'm going to run around the track 1 mile a day or whatever it is you're going to do. The first time you do that and you haven't done it in a while, how do you feel? Like hell. Okay. So, whatever. So then you go and you think, okay, I'm going to stick with this. I'm going to do it for 6 weeks. How do you feel 6 weeks out? Awesome. Okay. So that's because you have increased the number of mitochondria. You're more fit. You have the ability to use oxygen more efficiently. So oxygen, I mean, mitochondria rock. They are like amazing. And they're inherited only from your mother. You have lots of things to thank your mothers for. So the mitochondria only come from the mother. Why do you think that would be? That's weird. I mean, she's awesome, but really? Right? So why would they only come from the mom? Any ideas? Yeah.
Was it maybe because her egg being its own cell?
Yeah. The egg is huge. and the sperm are very small. So the egg, when the sperm fertilizes the egg, what does it contribute? I'm not denigrating males. But what does it contribute? Just the nucleus. So, you know, the DNA, basically. What does the mom contribute? Her cytoplasm in her egg cell sustains or the nutrients for the developing embryo. Is everything okay? Yeah,
Is that like in correlation with that, like, even if your mother was like, then for like more big or more like. Well, not how we're talking about like, having more big or more mitochondria, then like if your mother was more big, they have a higher chance of.
that is such an interesting question, and I don't think so. So, although I think it's more complicated than just saying no. But not in this kind of situation. If she had more mitochondria in her egg than it would really pass on to you. But for mitochondria being more important. Yeah. So mitochondria, the DNA in mitochondria sometimes is mutated and causes diseases. So sometimes there are diseases that are inherited with mitochondria as well. But basically, that's a little thumbnail of the mitochondria. The mitochondria have a special structure. They have two membranes. So they have a double membrane. This is an electron microscope picture of a mitochondrion. Mitochondrion is singular, mitochondria is plural. So this is the plasma membrane right here, or the inner membrane of the mitochondria, and this is the outer membrane. So this artist depiction is the inner membrane. The inner membrane has a lot of infoldings, which are called cristae. The infoldings increase the surface area for the attachment of proteins, which are also enzymes. So these proteins enable you to use oxygen to burn glucose. Essentially, you burn the fuel that you eat with oxygen to produce adequate amounts of ATP to fuel whatever needs your cells have. So this is where most of our ATP is produced, is in the mitochondria. They're little ATP factories. So you'll notice that some of the byproducts of the production of ATP, which is what your goal is, when you produce ATP, when you produce more ATP, you produce more heat. How does that relate to exercise?
Right.
Yeah, so as you use your skeletal muscles to run, for example, you're needing to have more energy. So your skeletal muscles are producing increased amounts of ATP and they heat up. They also produce a little bit of excess of a little bit of water. It's not going to make any difference to your fluids, really. It's only maybe, I think it's a few hundred milliliters a day. And then another byproduct is carbon dioxide. Question then question.
ATP is kind of heat, it causes heat. I guess my question is more related to like hormone, or like, we know, when we're going through menopause, we have hot flashes, but that's not like from too heat.
I don't think it's from ATP. I think it's more from blood vessel. And honestly, I don't know the answer. I'm not sure it's completely known why women get hot flashes, but I think it has to do with blood vessel, things going on, rather than ATP production per se. When you produce ATP, a byproduct of that is heat. So the conversion between the energy in glucose, for example, to ATP is not perfect. And so as you burn those molecules, some heat is released that is just a byproduct. Okay? It's not that it's not important, but your goal is to produce ATP, and in the meantime, you're producing. Yeah.
Is this also like, I don't actually understand this fully, but like when you lose weight, it's going out into that atmosphere, like you're breathing it out essentially, like, is that similar to this? Like, okay, you lose
I
weight, where does
wish.
it go? No, I mean, where does it go? Like, if you're exercising more,
Right,
it's like getting more mitochondria. And you're
Right.
producing heat, is that like another byproduct of it?
yes, I would say that's, your question has a lot of different levels, but I would say, I'm gonna give you the short version, which is that when you, so let's say you wanna lose weight, and one of the ways, what are the two ways that you make an effort to do that? Diet and exercise, right? You can take in decreased amount of energy is decreasing your calories, right? Or you can expend more energy. And that means that you're using more ATP. And if you're not eating as many calories, you're going to draw the energy from adipose tissue. So instead of, which is fat. So instead of using glucose that you eat for energy, you're going to use the stored energy that you can release from the lipid molecules that are stored in adipose tissue. So that's what shrinks the adipose tissue stores, and that's the weight loss. It's mostly adipose tissue. That's what you're hoping for. Anyway, I don't know that answered your question. Where does it go? You're right. You mean, because you're thinking of mass is neither lost nor,
Just like some
Created.
weird, yeah,
yeah,
yeah.
well, yeah, it's complicated. Maybe we'll answer more of those questions as we go. I hope. It's interesting. So these are other pictures of mitochondria, just showing the structure, that double membrane, the infoldings, which are called cristae. Anytime you see foldings like that, it means increased surface area. In this case, the increased surface area is for more enzymes to produce more ATP. And then we talked a little bit about the endoplasmic reticulum. The endoplasmic reticulum is membranes that are found mostly around the nucleus. It comes in two flavors, rough ER and smooth ER. Rough ER is rough because it has little buttons attached to it that are called ribosomes. So ribosomes are little structures that are associated with protein synthesis. So they're near the nucleus because the RNA comes right out to the ribosomes, and that is where the proteins are produced. All right. So I'm just going to show this one last picture. This is a picture of a cell. This is where the endoplasmic reticulum is found, right around the nucleus, and you have rough and smooth endoplasmic reticulum. And what I want to do now is stop for a moment, and we're going to have the opportunity to engage with these handouts that Chelsea prepared. So do you want to explain these a little bit? And let's just choose a section for people to do, maybe even the first page or.
They cover everything from the last week of this week's cell. So there's a lot to choose from.
So fascinating.
The first section is labeling parts of the cell. The second section is diagram forming components. The next page, I believe, is a list or part of the structure and function of organelles and cell parts.
So essentially, this is gonna be an opportunity for you to take like 15 minutes, so you cannot leave. If you leave, I'm gonna deduct points. So, so you're gonna take 15 minutes and you're gonna work together, and this is an opportunity. What I want you to do is to complete as much of the first page which is what we've just been going through. I want you to label it, I want you to ask questions of either Chelsea or me. So this is a great opportunity for you to do some studying. Work together, don't suffer alone. So use this time to use better to figure out the cards. Some of the analogies that you already mentioned, so did you hand this out to themselves? Did you hand these out?
Yes, Yep. They've got a head start. Yeah. These look defective because they don't have boxes. You can see this one does. This one does. I think Robin has the better one. Yeah. That staff doesn't have backs on the pages for some reason, but you guys can do the first page, I guess. Okay.
This is posted on your site.
They're in the announcements.
Okay, so yes, they're also posted on Canvas. In the announcements. Going in, because they read more.
Oh.
You're right. Yeah. Okay, so start talking to one another and figure out how to people.
That's fine. Yeah, I think that's fine.
So it was an odyssey. So what happened was, I totally got that. I really enjoyed that. Well, that's good. It's not.
I enjoy a good like mini emergency. That's like. That's great.
I was working on the stuff there.
Wow.
Yeah, this is, like I said, that first batch.
Is missing the. See each page, like the one that we did together.
Well, they should download it. Yeah. So you guys, if you didn't get the complete packet, you should download it from the announcement.
Should be double sized.
So there, it's in the announcements, do you know what I'm talking about? The Canvas announcements, Chelsea posted it. So if you want another copy to color or whatever, coloring really helps.
Oh, I guess it's not up yet. It
Okay,
will be
it
up.
will be
It
up.
will be up.
It
Either
will be
today or early tomorrow.
Okay,
Yeah.
so this.
And then I wanted to ask you about the wings. Should I go ahead and cover histology or Monday set? I would.
So, but I think I'm almost done
With this one.
Yeah.
Okay, so I'll just use the study guide to prepare for a histology session on Monday then.
Yeah.
And then
Yes.
Wednesday I'll
Yeah.
do just the test.
And when you come on Thursday? Yeah. Okay. Yeah. Cause I think it'd be great to have both of us, and I am gonna be running out of.
Okay, like right at the top, okay.
As soon as I can, I
Okay.
can't hang out, so I've got to make it manageable. So they're not stressed, so I'm thinking, question.
Yeah, that's what I was about.
I'm gonna get my children.
Oh, nice. Oh, yeah.
And then, I hope they're. Because they're prepared for
Yeah, we'll see. Usually there's a flood of students on plastic days. It's like when everyone comes out of the woodwork.
That's good.
Traditionally, at least in my experience.
So we should note that there is an athletic session
Yeah.
on Monday. So.
Monday will be Solving. And then Wednesday will be S review.
So this is, probably.
3:30 to 5.
Only for.
Yeah, you know, I think we never say it too much, just so they know it's an assigned do, but I send out. Yeah, lectures, and then the following Monday we'll keep it phone. Yeah. It is, especially if it's new to you. Which seemed me yesterday that it was. So.